We start off question 2. We have just finished question 1 on a different exam paper. This is, of course, A-level biology revision, paper 1, 9700. Today we look at, or this second question is from the 2024 paper, February-March. Remember, the topic is still microscope. So we want to look at our second question. The question says, a light microscope is used to observe two structures that are 20 nanometers apart on the slide. What is the actual distance between the two structures when the magnification is changed from times 40 to times 400? So you do your first reading just to get a general feel about the question. Then you look at it the second time, putting down the most important things that you notice. So number one, they've said that we're dealing with a light microscope. So you underline, it's a light microscope. We're not saying there's anything particular about it, but just so remember, it's not an electron microscope. And then they say they observe two structures that are 200 nanometers apart. Okay, now remember, when they're talking about structure, it means, think about, let's say you have two ribosomes, or a ribosome and a mitochondria, or a chloroplast and the nucleus. So we're saying the chloroplast, it could even be two chloroplasts that are 200 nanometers apart. All right. Okay, so that's, and then they say, what is the actual distance? So here we are focusing on the actual distance. So anytime you're thinking microscopy, you have to think, am I looking at the image distance or actual distance? So so the question is, what is the actual distance between the two structures? We already know that the distance is 200 nanometers, but it's 200 nanometers at what magnification? So they say, if you read on, the magnification is changed from times 40. So we are starting from times 40, and then it says to times 400. Okay, so we've magnified this magnification, this specimen, we have magnified it 10 times. Okay, so if you magnify 10 times, you get 400. All right, now question, what will be the actual distance? Now, to answer this question, you have to remember one thing. The actual size of a specimen does not really change when you increase the magnification. All right, remember, we have image size and actual size. Okay, so we could be having image distance, actual distance. So we're talking about actual distance here. So you may want to say that, hey, because we've magnified it 10 times, therefore we should multiply the distance apart 10 times. But this is almost like a trick question. The distance apart does not change. Okay? It does not change with an increase in magnification. Think about it this way. If you have an object that is, let's even say the nucleus is 5 or 1 micrometer, the actual size of that nucleus won't change even if you increase the magnification. The thing itself doesn't change in size. All right, the actual size of it. But if we asked you its image size, then the image size will change, but the actual won't change. So for this question, the answer is very straightforward once you remember that. But if you don't remember that, then it'll be difficult. So your answer therefore will be C, because even if I went to times 4,000, magnification does not affect the actual distance. All right, so however large or small, actual distance doesn't change. All right, so that's it to answer this question. So remember, we've said when you're asked about the actual distances, they're not affected by magnification. Okay, but image distance is affected by magnification. So that's it for question 2. We move on to question 3.